second presentation, I will be describing therapeutics that are in development in the pipeline for periodic paralysis. And again, my emphasis here is to give you a sense of the fact that there's a lot of activity going on now, and that this is not simply a situation where there's frustration and we're dealing with, managing risk factors and otherwise no other direction to possibly go. So you've had, I have no disclosures to make. You've had great introduction both yesterday afternoon and this morning by Dr. Sutherland about the cardinal features of periodic paralysis. And we really want to start there and from what's going on electrophysiologically, because that drives the strategies that were used to develop new therapeutics. So, you're dealing with episodic attacks of intermittent weakness caused by decrease or loss of excitability, common trigger factors of changes in temperature, stress, potassium, as you know. And something I'm going to touch upon, but we haven't discussed much at this meeting, is what patients are greatly concerned about is the slowly progressive fixed proximal myopathy that comes later in life. So the foundation for understanding all of this and what is in common with all forms of periodic paralysis in terms of the pathophysiology of the episodic attacks is here summarized in a computer simulation. Instead of having a normal hyperpolarized resting potential of about minus 90 millivolts from which the fiber is excitable, there's a shift during an ictal event of weakness in which the resting potential might be minus 50 or minus 40 millivolts, which of course inactivates sodium channels, causes the fiber to be refractory, completely analogous to depolarization block, such as what would occur with succinylcholine. So a lot of our strategic approaches were driven at thinking about how we could prevent that anomalous depolarization and thereby prevent these transient attacks of weakness. And you've seen this slide many times, and it's a wonderful way, I think, to put in context the fact that there are many channelopathies of skeletal muscle that impact excitability of the sarcolemma, that they fall on a clinical spectrum ranging from pure myotonia to pure episodic weakness and syndromes in between, that all of these are Mendelian disorders with mutations that change the function of ion channels expressed in skeletal muscle. And today I'm going to focus, not exclusively, but largely on those mutations that lead to susceptibility to hypokalemic periodic paralysis. And all of the studies that we do in terms of exploring new therapeutic opportunities are preclinical studies in animal models. So a little over a decade or so ago, we made a knock-in missense mutation in a mouse, the R528H, missense mutation in the calcium channel. This is the most commonly occurring cause of hypokalemic periodic paralysis. So the mouse looks fine, runs around the cage, similar to patients with periodic paralysis who most of the time are asymptomatic. And our outcome measure is the ex vivo contraction test. So this is a tetanic isometric contraction looking at peak force in normal potassium. And in this mouse, if the extracellular potassium in the organ bath is lower to 2 millimolar, there's a dramatic loss in force that does not occur in normal muscle. So here's a summary dose response that clinical neurophysiologists would love to have for their patients, but it's impossible to do in vivo, which is to really understand the sensitivity for the loss of force as a function of extracellular potassium. So here in the open circles are the wild types, so they tolerate an amazingly wide variation in extracellular potassium in this ex vivo contraction test, as opposed to these mice with susceptibility clearly to hypo, but not hyperkalemic periodic paralysis. Interestingly, these are a little gene dosage effect. The filled symbols are homozygous mice. You can breed them to homozygosity. They survive. What's not shown here, this is all males, so similar to what Dr. Sutherland told you last hour, this is a really high fidelity model of periodic paralysis in humans because the female mice are less susceptible to these attacks, just like the humans. So it's really a spectacular model. So for the rest of the morning, what I'm going to be doing is thinking about how we can use this system to test strategies for preventing this anomalous depolarization and loss of excitability. And this cartoon, this cylinder, represents the myofiber, and the relevant players that are helping to set the resting potential, and it's more than just the baseline potassium conductance. There's an important chloride conductance, sodium potassium ATPase pump, and a co-transporter. And the first approach you might take, and indeed when the field of channelopathies first evolved in the early 1990s, the idea was, okay, you've identified missense mutation of pathogenic channels and characterize how their behavior is altered. Why not look for small molecules drugs that could normalize that behavior? So the target is the misbehaving channel. So there's some challenges with that. These are a summary of the mutations that occur in human hypokalemic periodic paralysis. And as you've heard a couple of times, the discovery was initially made in the calcium channel. And in the early 90s, the world was simple. If you had a calcium channel missense mutation, you were at risk of hypokalemic periodic paralysis. But the sodium channel mutations, it was hyperkalemic or paramyotonia congenita and myotonia. That pattern was later revised because families with absolutely classical hypokalemic periodic paralysis that to experts could not clinically distinguish a difference on the basis of the nature of the attacks or the long exercise tests or anything else, happened to have missense mutations in the sodium channel. And it took a long time before physiologists understood why these mutations altered the functions of channel to put the patients at risk for hypo-PP. But in hindsight, when you can look at this now, something like 24 out of 25 of these missense mutations all start with the letter R because they are all at arginine mutations in the voltage sensor domain of the channel. So there's no way that is by coincidence. That is a fundamental feature of the change in the channel protein that has these individuals susceptible to attacks of weakness. And what we and others have learned is that these specific mutations, again, in the part of the channel that gives it its sensitivity to membrane potential and enables voltage-activated channels to open and close and inactivate in response to voltage, what happens is it doesn't alter those properties per se, except for the one R222 you heard about this prior talk. What happens is when there is the wrong amino acid in this voltage sensor, a missense mutation, the physical association between that sensor and the rest of the channel is disrupted, and it creates a leak pathway where literally there can be a small, albeit feeble, current that is conducted in this anomalous pathway through mutant channels. And that's why mutations in either sodium or calcium channels can lead to the same clinical phenotype, because there's conservation evolutionarily of this voltage sensor domain. It uses the same body plan. So if you have a disruption in that body plan, it doesn't matter whether you're a sodium or a calcium channel, you'll have the same leak and the same susceptibility to periodic paralysis. So the idea was, let's find a drug that plugs the leak. But the challenge is, while there's a rich history of the pharmacology of channel blockers, greater than 60 years, those are all acting at the conventional ion-conducting pore. None of them are relevant to this anomalous pathway. So there were no leads upon which we could start this investigation. And what turned out is there is another channel that conducts protons, it acidifies organelles inside cells. These are the voltage-dependent proton channels. And just their natural body plan, they are missing the ion-conducting pore, but they have voltage sensors. So their voltage sensors somehow also conduct protons, which is what happens in some of the leak cases in periodic paralysis. So we paired up with Francesco Tombola, who's at UC Irvine, who had been studying those channels, and by using a combination of high-throughput screens and molecular dynamics of looking at the crystal structure of these channels, recognized that there were small molecules that could block these channels. And the molecules had little features on them that looked a lot like the structure of arginine, which is the amino acid that's supposed to be there. Cut to the chase, he shared some of these compounds with us, and we tested it in our hypo-PP mouse. So this is the calcium channel mutant mouse. What we're doing is the ex vivo contraction test, and you're looking at normalized force starting at 100%, and once every 2 minutes, we're measuring the maximum isometric contraction of this muscle from a mouse susceptible to hypo-PP. So in the black traces, when the potassium is decreased, there's the 50% loss of force that recovers when you return the potassium. If you pre-treat these animals with this small compound, you can attenuate that loss of force by nearly 50%. So there's great promise there. In comparison, in green is the response in wild-type mice. We're still working on optimizing these, and it's an ongoing study, but the concept of why don't you just plug the leak, it would be the silver bullet to ameliorate these episodic attacks of weakness and hypo-PP. So there are other strategies. It turns out a general theme that's coming to all channelopathies is instead of targeting the mutant channel itself, you can look at other players that influence the resting potential or the electrical excitability of the cell. And a wonderful target that was recognized by electrophysiologists 30 years ago, Frank Lehman-Horn, Reinhard Rudel, and all them, would be if you could increase the potassium conductance of a muscle fiber, then you would promote hyperpolarization. Again, here's our action potential in an excitable fiber and the depolarized shift that renders the fiber inexcitable. Well, the potassium gradient sets a reversal potential that's very negative. So if you could make this cell more favorable to conducting potassium, you would tend to keep the membrane potential closer to EK, and that should work. So there are a number of different potassium channels under which this is possible. And the one we studied most recently is a drug that was initially marketed as an anti-epileptic, retigabine. And what I'm showing you here, this is not a computer simulation, this is a recording over an hour or so of just the resting potential of a muscle fiber from a mouse with hypo-PP. And so here's the provocative, this is the sodium channel cause of hypo-PP. When we lower the extracellular potassium in the bath, instead of hyperpolarizing, which is what normal muscle should do, this muscle depolarizes. So that's a wrong way change in the voltage. So the magenta highlight is the anomaly. When you return the potassium to a normal value, the fiber repolarizes. If you treat with retigabine, which opens the KCNQ family of channels, of potassium channels, it completely prevents the anomalous depolarization. And if you look at force as the outcome measure, this is again returning to the calcium channel hypo-PP mouse. Now what we're doing, this is sort of an N of one, where the one mouse is its own control. So we study the soleus muscle in one dish with no drug and look at an induced attack. And in the other organ bath, a muscle from the same animal is being treated with retigabine. And here we're looking at a rescue experiment. So initially the potassium is reduced to 2 millimolar. There's a dramatic loss of force on both muscle preparations. And then when retigabine is added on top, you can see there's a recovery or a rescue showing that it's possible that this could be used in an abortive therapy context, which of course is really important in the management of periodic paralysis because patients don't like to have to take prophylactic medication every single day. We've done other studies, again, looking at prophylaxis. This is now with the sodium channel hypo-PP. It turns out these mice have spontaneous oscillations during the stress of low potassium to trigger an event, so they lose force and spontaneously recover and lose force again. So here's one example, but if we pre-treat with retigabine, the whole episode is prevented. Here's the dose-response relationship looking in the blue symbols for the sodium channel-based hypo-PP mouse model, and in red, the calcium channel. And you can see with low micromolar concentrations of retigabine, completely obtainable therapeutically in patients, you can dramatically prevent the drop in force. And this is about 10-fold more potent than prior attempts with potassium channel openers using panacedyl, which opens KATP-sensitive channels. The challenge there is those KATP-sensitive channels are in smooth muscle, and so they relax arterioles, and people would have hypotension and faint. So panacedyl ended up not being a good option as a potassium channel opener to prevent or abort attacks of periodic paralysis. But this shows, in my opinion, fantastic potential. We've also looked at hyper-PP. This was an N of 1. It just happened to be one of those days. Again, each mouse is its own internal control. This had sort of a strange response where the onset of weakness was late and not as large as we usually see. There was some protection. It was not as dramatic. We're still working that out, but I think there's a possibility that hyper-PP would benefit from this as well. We also have a pharmacologic model for Anderson-Tawil syndrome by blocking the inward rectifier potassium channel. And that's a complex potassium-sensitive form of periodic paralysis where either a reduction or an increase in potassium can trigger a decrease in force. And what you can see is pre-treatment with retigabine, again, is extraordinarily effective at preventing the loss of force from a low K challenge and offers some protection from the loss of force in a high potassium challenge. So we're optimistic that this strategy, and particularly agonist for this class of potassium channels, will be applicable widely across many forms of periodic paralysis. So many of you may know that retigabine was FDA approved as adjuvant for partial epilepsy in 2011, but then was taken off the market because there was a bluish discoloration of skin, some hepatic toxicity and mild CNS effects, and the market just wouldn't tolerate that level of adverse events. So you might think, well, nice study in animals, but this is irrelevant to be able to help patients. But the pharmaceutical industry remains very enthusiastic about the concept of potassium channel opener drugs as a class for any disorder with hyperexcitability, epilepsy, arrhythmias, periodic paralysis. And Xenon Pharmaceuticals, which I have no connection with at all, is developing derivatives that open the same class of potassium channels that retigabine does, but does not cause the blue discoloration or some of the other problems. In fact, that's in phase 2 trials, as you'll see in a minute. And again, we do not have an affiliation with Xenon Pharmaceuticals, but we looked at the patent and found the chemical structure of XEN 1101, and it spectacularly protects muscle in our mouse model of hypo-PP from an episode of weakness. And just last month, part of this phase 2B study of using XEN 1101 came out in Archives of Neurology. So this is really close to being out there and available for management of epilepsy, and will be an opportunity for off-label use, I think, for periodic paralysis. So again, as many of you who know me, we work primarily with preclinical models in animals. I'm hoping our clinical colleagues will do a well-controlled crossover study here so that we really get high-quality data for this as a class of medication, because I think there's a wonderful potential here. So one more topic to go through on, which is gene therapy, which of course is a hot topic and approach for many neuromuscular disorders. But I want to point out a couple of features of periodic paralysis for why this makes sense. So what I'm showing here schematically is a sort of burden of disease, disability, episodic weakness over the lifespan. So starting out in adolescence and childhood are dramatic episodic attacks with complete recovery. With age, these tend to become less severe in terms of their intensity, but sometimes prolonged in duration, and then become replaced with this slowly progressive fixed myopathy. And the pathophysiologic basis for this is a little unclear. Its relation to the episodic attacks is still something that's under investigation. But you can see where this would be really difficult to ever have some type of controlled trial. You'd need compliance for decades to see if you're going to make an impact on the development of disease. And so a traditional pharmacologic approach is really a challenge here, whereas gene therapy with intervention early in life would offer an opportunity. So let's talk about what this might look like in periodic paralysis. So one concept, it would be to repair the defect. So in almost every case of periodic paralysis, both hypo and hyper, these are missense mutations that caused by a single nucleotide transversion. There are a couple with small deletions, but those are incredibly rare. So that means there's an opportunity for these modified CRISPR-Cas proteins that become if you will, editors and correctors rather than just making a double-strand break in the DNA and destroying the allele. You can actually make corrections. So we've explored the possibility to do this starting from our mouse models of periodic paralysis. So we start with a missense mutation either in the calcium channel or the sodium channel, and then try to correct it in the cells in dish. So it's one better than in the test tube with working, completely just with enzymes, so actually in live cells. And so what you're looking at here, this is how sequencing data comes back to you looking at the standard Sanger sequencing of the chromatogram, and you can just read the base pairs off here. And the point I want to make is we've done this both in the calcium channel R528H mouse and in the second most common cause of hyper-PP, we have another mouse. So here you can see what is an A in the mutant switching back to a G. It's not with 100% efficiency, but we had 34% correction able to do in these myoblasts and cultures, and a little bit higher percentage in the hyper-PP mouse. Still a ways to go. One of the challenges of this technology is you use a guide RNA to direct your base editor to where to make the correction, and you could have bystander editing that you don't mean to have happen, which occurred here. So there's some fine-tuning that has to happen, but this is promising and is an area of under development in our lab. The other approach, which wouldn't work for every Mendelian disorder, but is particularly relevant for periodic paralysis, is to ablate or destroy the mutant copy of the gene. So why is that possible? So in all cases, it's altered function or gain of function for sodium channels and calcium channels that causes periodic paralysis. So it's if you could selectively destroy, you have two copies of each of these ion channel genes, one from mom and one from dad. If you could selectively destroy the mutant allele with a CRISPR-Cas9 by making a double-strand break and random insertions and deletions, that might be a strategy. So you would lose this dominant negative effect, and there's always the concern with this technology, the so-called insertions and deletions to destroy an allele. What if you are unlucky and get a multiple of 3, the triplet of the codon? Then you'll make a Frankenstein protein, right? You'll introduce a new mutation and could create another disease. But here, again, for these forms of periodic paralysis, we're fortunate because this region of the protein where you have one of these charged arginines, every third amino acid, does not tolerate a deletion or an insertion in frame. Those channels have been studied and they don't function. So we're safe in that those types of so-called ablations would not cause a problem in this case. There's been the question, why don't you look at interfering RNAs instead of using CRISPR, which is dangerous, editing genes in patients? But you'd have to then have these, interfering RNA therapeutics administered lifelong, and addressing, will you make an impact on the permanent muscle weakness becomes more challenging. These are the same papers that Dr. Sutherland showed you, actually, and what it demonstrates is in large cohort studies looking at things like unusual cases of congenital myopathy, where the power of whole exome sequencing now has identified cases, rare cases where this happens at ion channels that are also involved in periodic paralysis. And what's interesting is in both of these situations, for the sodium channel and the calcium channel, the phenotype, the myopathy, was recessive. And the parents are asymptomatic, and some of those mutations they have are functional null alleles. So there are already established examples in humans where you are just fine with one normal copy of the gene. So again, it says you could ablate the mutant allele as a strategy. So we've done that. And we're now using these CRISPR-Cas9 ablation technologies in mice, not just in the culture in the dish. So the big challenge there, any of you who are following the Duchenne dystrophy or SMA and all that, is the delivery of these tools to the tissue with adeno-associated viruses or lipid nanoparticles. This is a huge challenge in muscle disorders. So for proof of principle in this preclinical study, you can locally deliver these genetic instructions to do CRISPR editing in muscle, in a live mouse, by just injecting the plasmid DNA in the footpad of the mouse and under anesthesia apply an electric shock, which transiently disrupts the membrane of the muscle fibers. They take up the plasmid, and you can do gene delivery focally that way. So here's an example of how that worked, because in addition to the gene editing machinery, we put in the code for the green fluorescent protein. So here is just a light micrograph of a dissected foot pad of the mouse. So you're looking at the interosseous muscles of the hindfoot of a mouse under bright light field. Under fluorescence light, you can see all the green muscle fibers are where the marker is being expressed. And in fact, we can enzymatically dissociate the mouse so that all the fibers fall apart, and you can pick up individual green fibers where you know all the machinery should be there because you successfully got it in there, and then look at your efficiency of editing. So again, you sequence the DNA. There's a mixture, a gamish of different types of insertions and deletions, and some were edited and some were not. And so what happens is the DNA sequencing is all clean. You got a nice sequence at our target. The blue box is where the CRISPR is supposed to make its cut. And downstream from that cut, the sequence is all jumbled up in a mess because there are random insertions and deletions, and you're looking at a whole population of different genome sequences from these muscle fibers. This is summarized in a histogram where you can use computational methods to sort of untangle this. And the bottom line is about 50% of the target genes were disrupted. So half of them are normal, and all of the disruptions ended up being deletions of either 1, 2, 3, or 4 nucleotides. In comparison, we did this exact same experiment in a wild-type mouse, and none of the wild-type gene was disrupted. So here's the selectivity, specificity for only editing the mutant allele. So the really exciting part to share with you is 4 weeks after doing this, how well does the muscle perform? So what we do is a control. One side, one foot pad is injected with all the machinery to do the gene editing. The other is missing some pieces. So it's either just the guide RNA or just the CRISPR, but not the whole package. And what you can see, even at baseline in normal potassium from this hypo-PP mouse, is that there's a difference in just the baseline maximal tetanic contraction of the flexor digitorum brevis, even in normal potassium. And if you look at a whole population, in general for this model, even in normal potassium for this sodium channel model of hypo-PP, there's a clear reduction in the baseline force that on average was improved by doing the gene editing. But was even more impressive is when you give the potassium challenge. So here's the additional loss of force in the susceptible muscle. Again, this is muscle from the very same animal, but where the gene editing machinery was electroporated into the muscle, and you've protected that muscle from the loss of force. And here's an example where instead of looking at individual force traces, we've now done this assay again of the low potassium challenge over 90 minutes, where every 2 minutes we're measuring the baseline isometric contraction, the low potassium challenge, and then return potassium to normal, so you can see that there is the spontaneous recovery. But comparing one side where the gene editing occurred to the other, there was a dramatic difference, and we've now done this 5 times with slightly different constructs all targeting the same site. And you know, most of us like to show average and standard error of the mean because there's some outliers and it's messy, but I'm showing every single individual mouse because each time this gene editing was successful at protecting the muscle from an induced episode of weakness with a low potassium challenge. So we're really excited about this. A long way to go. We just got a lot of new resources from the Muscular Dystrophy Association, the NIH, Periodic Paralysis Association, to take this work further, which we're really excited about. So I just want to finish with a concept about gene editing and how good you have to be and how realistic is this beyond the question of delivery to muscle, which is a huge challenge. But what we're really excited about is, again, the motivation is there to try to make some inroads on the risk for late proximal myopathy. And again, gene editing in these channelopathies has several advantages. So in addition to looking at the genomic DNA with next generation sequencing and seeing how efficient were your molecular tools at making the changes to the genes, for a channelopathy, you can then measure the ionic currents in those muscle fibers, and you can explicitly, quantitatively measure the impact of your editing, because it's never going to be 100%, and you can measure it exquisitely, precisely, more than other genetic diseases. And then we can understand with our animal models just what percent editing do you need to achieve in order to have an impact on the clinical outcome. And we can do that in our animal models, and also, just as James Fraser does, we have models for excitability of muscle fiber. And we have shown, for example, that when you lower the potassium, you should normally hyperpolarize. That's the normal response. If you have this model simulate hypo-PP, you inappropriately depolarize, and then in silico, in the model, you can begin tweaking the amount of editing you're simulating. And what the model says correlates with the model, with the actual experiment in the mice, that something on the order of 30% editing is all you need. And that's very important for feasibility and later translation of this technology, because if you had to get every last mutant allele either modified or deleted, the technical challenges would be much larger. So just finish with this summary slide that we're using a number of different approaches, all to tackle the same problem of instability of the resting potential. You could use pharmacologic approaches for the actual leaky channels. Certainly could be a benefit for the episodic attacks, and maybe even for risk of late permanent weakness, because you're removing the inciting event. These other tricks at keeping the fiber hyperpolarized, either from opening potassium channels, or I didn't mention it today, but there's another technique involved in manipulating the chloride gradient. That's sort of a secondary way of stabilizing the resting potential. I still think these are both excellent choices for management of symptoms and acute episodes of weakness, but probably not as optimistic about what they might do for risk of permanent weakness. And then, of course, gene editing would be the gold standard to try to prevent both of those. So just finish up, I'd like to thank, we have a small lab and some really talented individuals. Fen Wu does all the gene editing manipulations. Marbella Quinones does all of the muscle force recordings. Her husband, Marino DeFranco, is just an incredibly skilled electrophysiologist, made some of those challenging recordings. My wife's lab at UCLA is interested in limb girdle dystrophies and mouse models and gene therapy, and she really brought us electrophysiologists into this whole world of gene editing and how to do this that we're very grateful for. We've had some support, as I mentioned, from the NIH, from the Muscular Dystrophy Association, the Broad Institute of Stem Cells at UCLA, and the Periodic Paralysis Association. So thank you very much, and I'm happy to answer any questions.
So I've read your papers with about 12 different highlighters, and I still don't understand how the calcium channel defect produces the weakness. So in a paragraph or so, can you handle that?
Sure. Here's the clicker. So the question is, in case it didn't get on the recorded video, is the pathophysiology of why a leaky calcium channel puts you at risk of low potassium-induced weakness. That's the question. And why do you get the wrong way change in the resting potential? Especially if the leak is there all the time. Why does it? And I promised this was not a plant, but I have a slide about that. So here's the mouse model. Here we can do CMAPs actually in vivo. So here's the figure that your highlighter ran out of ink on. And the basic principle, what you're looking at here is membrane potential on the horizontal axis, what would be the resting potential, and the currents that are flowing, the ionic currents that establish that. And in all of this complicated squiggly lines, the important one is where the black line crosses the horizontal axis, because this is the total currents added together, and these are all the individual ones. It's where the total current is 0 is what sets the resting potential. Because if there is any current means charge coming in or out of the cell, that will change the voltage. So if you don't want the voltage to change, your total current has to be 0. And what you can see, it's not just the simple Nernst equation for potassium, which is what you learn in college or medical school, that there are a lot of different players. Here's the window current that Dr. Sutherland was mentioning. This is a big player. This KIR is the inward rectifier potassium current. So all of the currents that normally, if you're not at the resting potential, would be flowing and are negative numbers, that's charges coming in that would depolarize the fiber. So anything on the bottom half here would cause your fiber to want to depolarize. On the top half would help hyperpolarize. And what's happening, it's a tug of war between these. And what the gating pore leak does in the mutant channels, that's this little red line. So you get an additional tendency to depolarize because you have an additional leak. And that's okay. You can compensate that for that when your potassium is normal. But in all of us, you, me, and everybody else, when the potassium outside of cells is reduced, this relationship, this contribution from the inward rectifier potassium conductance, the same one that's responsible for Anderson-Tawil syndrome, this shifts to the left. And is it because of the change in the potassium equilibrium potential? And it shifts to the left and changes in amplitude. So what happens is you have a potassium-sensitive modification or tweak to these balancing currents. And what was initially a very small leak that's the problem and was of no consequence now causes a problem because your net balance of currents becomes negative at the voltage where it should be, and the only way you can stop the process is depolarize and turn on other potassium channels. So it's a combination of having a new risk that would cause you to depolarize, but you tolerate that risk with normal levels of potassium. When potassium is low, by a mechanism that operates in all of us, that shift we tolerate, but these individuals don't, because this extra burden now manifests itself as an influence that will depolarize the cell. That's probably more than you wanted, but we can talk afterwards as well.
Thank you.
Can I ask two questions, if that's all right? Firstly, with the sodium channel mouse and the oscillations, they're sort of quite fascinating as well, given the time scale of them. And I just wondered if you could comment on what you think's going on.
Yes, so a great question. So interestingly, while there's a provocative challenge of low potassium that we are controlling, it's in the organ bath. This is not an animal with some kind of homeostatic compensation at the whole organism level. These oscillations happen almost exclusively in the sodium channel-based model of hypo-PP, not in the calcium. They're very slow. They take minutes, and they will go, we've done it for 2 hours, and it keeps going. Our best guess at what's going on is, of course, when this happens and there's the depolarization, and it happens in the resting, you can see it in the resting potential as well, that when it's depolarized and there are a lot of ion shifts, a lot of sodium comes in, that's a huge stimulus to increase the activity of the sodium-potassium ATPase, which takes minutes to occur. The upregulation of the pump rescues you, then the pump goes back to its baseline, whoops, then you're going to have and you're cycling back and forth. So it's a terrific model for how does spontaneous recovery occur, which is something that has not been very well addressed for the acute attacks. So it's a wonderful opportunity to study in greater detail. The other thing that is just amazing, I don't have an example here, but we always study the soleus muscles from the left and the right side in parallel in the same experiment. The oscillations are slightly different from prep to prep, but when you look at these two muscles in two completely different organ baths, no connection whatsoever, the oscillations are exactly in phase. So they're, just like patients know, there is some set point before we extract the muscles that's shifting on a slow time scale. So patients have a period where I've had a really tough, difficult few months with a lot of attacks or not. And so there are interesting things going on that's modulating the susceptibility. And it's clearly at a systemic level because the two muscles are exactly the same in two separate organs. It's fascinating. So you could tell I can get excited about these things. I think there's time if you wanted your other.
So my other question is whether or not, it's a slightly selfish question, but whether you looked at supernormality in your model and whether it exists.
Yeah, so For supernormality, so my hat's off to James Fraser, because it's one thing to get these accurate models of risk of depolarization and myotonic discharges in an isolated model of a muscle. When you include the fiber and a conduction process, oh my gosh, the computational cost is astronomical. So he'll be a better choice for that. But I would love to invite you to Los Angeles and measure velocity recovery curves in these mice. Absolutely.
Thank you. Wonderful talk. And every time, I'm still trying to analyze and understand that, but at some point will. So as you said, one of the main issues is delivery. And, even the focal electroporation you did was 50%, I think, successful, correct? So what would you think in terms of whole body delivery? And that's always a challenge. I know it's not, but that's something we eventually have to get to. So what are your thoughts about it in general?
It's a great point, and there are multiple steps at which the efficiency is not 100%. So just to be clear, first, delivering these gene editing tools, even by the electroporation technique, was not 100%. So that's why we separated out just the green fibers and did the genomic analysis only on the green fibers. Even when we looked only at green fibers, the editing was only 50%. So those were 100% transfected, but only 50% edited. And so at every stage, you lose a little, The good news is I'm confident you're not going to need 100%. Again, my wife, Melissa Spencer, does a lot of this for Duchenne. They have a CRISPR strategy for doing in-frame deletions and having truncated dystrophin. And what they are getting with their best efforts in AAV, adeno-associated viral models in mice, is, 2 to 5% recovery of dystrophin. Now, that's after many, many steps. So where the problem is unknown. We've looked at this in many different ways. There's a California Nanosystems Technology Center at UCLA, which does a lot of lipid nanoparticle and custom encapsulation. The thing is, there's a really tough extracellular matrix basement membrane on muscle fibers, as there must be to withstand the mechanical load. And that's a huge physical barrier for a lot of these technologies to deliver these products. And someday we'll get there, but it's going to take a lot of work.
Thank you. But really fascinating. Thank you.
Thanks. All right, we stayed on time. Thank you very much. And I think the Periodic Paralysis Association is going to make these available later on their website, if you'd like to review. Thank you very much.
If you would like to know more about periodic paralysis, visit periodicparalysis.org. And if you enjoyed this video and want more, hit that like button, subscribe to our channel, and hit that bell so you don't miss any future videos. It really does help spread the word. You can view other videos about periodic paralysis by clicking the thumbnails to the right. If you have questions, just leave a comment below or reach out to us on social media. We'd love to hear from you.